thank you, Gary, and thank you, Joyce, and the Vasculitis Foundation for hosting a wonderful conference and for inviting me to speak yesterday and today. I'm going to start out by saying I get it. I know what most of you are thinking, and that is you look up on that screen and you see biomarkers and you see translational research and you say, oh no, it's the last day of a conference, my head's about to explode, and I don't know what those words mean, but I know science is coming at me. So I promise you, the goal and the objective of this talk is to not focus so much on the specifics. Actually, the main objective is to get you excited about the future, about what we're doing right now, and to empower you so that you understand the process of how we're thinking and the directions that we're going. Because if you're empowered, I think you'll be eager to participate in this research, and that's one of the big hopes of this talk. There are some other objectives, of course, the more specific nuts and bolts of the talk, which is that I know I need to define for you what a biomarker is and what translational research means. And I really want to focus on how new technologies are influencing biomarker discovery. And that's a key word in this talk, is discovery. And that's really the exciting aspect of it, because we're learning new things. And then finally, I'll touch on this a little bit, and I'll review some of the biomarker discovery that's happened in vasculitis. But I'm not going to, this will by no means be comprehensive. And when most people get up and say, by no means comprehensive, that usually means they're only going to talk about their own work, which is probably what I'm going to do. Although not entirely. So let's start with the word biomarker. So a definition of biomarker is that it's a measurable characteristic that reflects information about a state of disease or health. And let me put this into a more concrete aspect for you. So I wish I could say that this was a story of the olden days, but actually this is a story of the present. This is what we do now. And let's say that you have vasculitis and your disease has been controlled for a number of years, and you're doing great. You're off your medications. Maybe your doctor spaced out your visits. Instead of seeing you every 3 months, you're seen every 6 months. And then all of a sudden, you call your doctor and you say, I need to come in. You need to see me. And you come in and your doctor says, what's going on? And you lift up your pant legs and show him this. Now, I know many of you probably experienced this, but for those of you that haven't, this is a medical term called palpable purpura. It's inflammation of the blood vessels in the skin. This is a form of vasculitis. And your doctor takes a look at this and says, oh no, your vasculitis is back. You had a relapse. You're flaring. And this is the way we do things right now. We kind of, in some extent, we wait for things to happen. We watch very closely. We monitor everything that we can. And we just let nature take its course. And if something happens, we react as quickly as possible. And this isn't really the standard that we should hold ourselves to. We need to be able to know what's coming. We need to be able to predict what's happening down the road. We need to be able to tell you at the onset of your disease what your most likely outcome is going to be. And we need to be able to tailor our treatments to those predictions. These are toxic medications that we use that have side effects. It would be really nice if we had biomarkers, if we had things we could measure that would guide our therapeutic decisions. It would make our lives easier, and it would make your lives much easier. So with that in mind, this is kind of the goal, the over outline of what we're doing with biomarkers. So one way that you can think about it is to define risk. You want to identify patients who are likely to develop disease. And some of this ties in with Sharon Chung's talk about genetics. So it would be nice if we could look into your genes and see, are you at risk for developing these diseases or not? And as she outlined for you, there's challenges in doing that with vasculitis, because a lot of the genes that are associated with vasculitis only confer a really small, tiny increased risk, and a lot of healthy people that never develop vasculitis have these genes. But that's one goal. Another goal would be diagnosis. Can we detect your disease earlier? It's a common theme among patients with vasculitis that you go months and years and you see many, many different doctors before you get a diagnosis. And in part, that reflects a deficiency on our part. We need better tests. We need to be able to give your primary care physician an easier test to make the diagnosis earlier. There's also ways to classify the disease. You know, if you gather a group of patients together, and the nice thing is you can meet patients at these symposiums with similar disease, and you can share your stories. And you'll notice that there are some similarities. There are a lot of similarities, but there are also differences. Maybe in the table with patients with GPA, some of you have had just nose and airway involvement, and some of you have had kidney involvement. Some of you have had both. So clearly there are different subgroups within these diseases. How can we better define those and distinguish between those? Characterization of response to treatment. How do we know if the medications are working? A lot of this is trial and error. We give you a medicine, we watch you, and it sometimes can take many months to determine if that medication is effective or not. Can we measure something in the blood sooner that will let us know? So we can, if the medicine doesn't work, we can switch courses earlier. And then prognosis. And I think prognosis is probably the most important thing. And I would say once you have these diseases for a while and you're comfortable with your diagnosis and you know what your diagnosis is, that no longer becomes so important to you. What's important is what's going to happen to you over time. How is your disease going to behave? Are you going to have multiple relapses? Or are things going to be stable and quiet? This is a key concern for most patients with vasculitis once you get over the initial phase and shock of being diagnosed with the disease. So can we find markers in the blood that predict how your disease is going to progress, that predict who's going to relapse and who is not going to relapse? Can we identify patients who are likely to respond to specific therapies? These are the goals. And we're entering the age of omics. And I don't know how many of you are familiar with this, but if you start looking in medical literature these days, you see terms like genomics, transcriptomics, proteomics, metabolomics. They're daunting terms, they're confusing terms, but at the heart of them is technology. And so I wanted to take a few moments to kind of explain to you how technology is influencing and shaping some of the ways that we do research. And that is that we are now able to do things called high throughput technology, which means we can measure thousands and thousands of things simultaneously. And we can analyze those things with computers in ways that we couldn't do 10, 15 years ago. So for example, wouldn't it be nice to know what's happening inside of a cell? And when we used to do it in the past, we'd pick a specific target, we'd measure that and see what's going on. Now, we know that there are 27,000 genes in the human body. And that actually is not a lot. It's much less than we originally thought. There are 27,000 genes. We can measure them all at once within a group of cells. And then we can take patients with disease A and disease B, measure all 27,000 genes, and compare them. And this leads to big data, and that's the age that we're entering into. Graduate students are coming in to analyze projects, and they're surprised if they're not getting gigabytes and terabytes of data. I mean, these are the challenges that we're facing, is how do we interpret all of this large amount of information that we're generating? And I think we're right on the edge of being able to do it really successfully, and that's what makes me really, really excited about what's to come. And it's kind of paradigm shifting, you know. In the past, science and the scientific method worked by, you had a brilliant idea, you read the literature on a topic, you understood it, and you came up with your own idea related to that literature, and then you tested it, and you saw if it worked or not. And now we're kind of getting into technologies that have allowed us to be a little bit more focused on discovery, where you don't necessarily have to have a concrete idea. You can generate large amounts of data and let the data speak to you. A lot of people, and that's problematic in many ways. It creates issues where you can discover things that aren't really true just by the fact that you're looking at so many different things. That's called false discovery. And there are a lot of critics of discovery-based methods that think you're kind of wandering around blindly in the dark. But I tend to look at it like this. Let me go. So this is a picture of my family at the zoo. And you'll notice, if you look where the focus of the picture is, there's my wife and my 1-year-old daughter, Nina, having a great time. But if you look in the top right corner of the picture, you'll see my other daughter, Riley. She's 2 years old. She's in the process of taking off her pants. And this is a tidal pool at the zoo, so this is water that comes flooding in, and I'm standing on one side of it, and right as I see this, the water rushes in. I can't get to her. I have shoes and long pants on, so I have to just watch and see what happens, and she proceeds to get entirely naked. And when I finally got to her, she said, "That's what you do at the zoo, Dad. get naked. The point of this slide, I'm not sure what the point is, but I thought it was funny. Now, the point of this slide is that if we keep looking in the same places, if we keep looking where we think things are, if we look where the light is, we're not going to notice what's going on in the periphery. And this is termed a streetlight effect, where, you know, if you're always looking where everybody else is looking, you're not going to see what's going on in the rest of the parking lot. So one small area of the parking lot lit up by the street lamp, but there could be a lot of stuff going on in the periphery, and we're starting to realize with these technologies that the parking lot is miles and miles and miles long, and it starts to make looking in these one little spots where everybody's looking more and more foolish. So this is the light. This is where the light is currently. This is what we use as biomarkers in vasculitis. There's a lot of other things that are biomarkers in vasculitis, but these are the main ones that we look at clinically. So we look at something called the erythrocyte sedimentation rate, the ESR, and the C-reactive protein, the CRP. These are markers of inflammation. And I'm sure for many of you, these markers have driven you crazy. And if they've driven you crazy, I can also testify they drive us crazy too. The reason is because they can be elevated, and when you have inflammation, they can be an indicator that the disease is active. But also, they can be elevated and there's no disease. Or they can be normal and your disease can be active. So you can drive yourself crazy really thinking about these markers, and really what that means to us is that they're not good enough. They're not reliable enough to make treatment decisions. We need better things. We also look at your white blood cell count, we look at your hemoglobin, we look at your platelet. These are all indirect measures of inflammation and how things are going on. We look in your urine for blood and protein. The ANCA test, so if those of you with GPA and MPA and eGPA familiar with this test, it's a test that's very useful to diagnose these diseases, but when you start trying to use this test to figure out how things are going over time, it becomes much more problematic. And then eosinophil levels in Church-Strauss and eGPA, that's another thing that we follow. But we know from experience that these are imperfect markers. They're not giving us good enough information. We need better ones. And so the process by which we're finding these better ones is a process called translational research. Now, within research, there's a lot of different branches. There's clinical research, where you have doctors and patients working together, maybe clinical trials where you're testing an experimental drug, or you're measuring things in patients over time and kind of seeing how that relates. And then there's another group of basic scientists. These are people in the labs that are working with mouse models of disease, working with petri dish plates with cells, and they're looking at pathways and molecules of interest. And then there's a third group, I think, that's germane, and particularly so in these days, which is the analytic people, the people that can do the statistical measuring and the statistical work. And in the past, a lot of this research has happened, independently. And what we really need is to link these people. And that's at the core of translational research. It's actually bringing basic scientists, bringing clinicians, bringing statisticians to the table so that we can design and implement effective strategies for research. And more importantly than that is this concept. What drives translational research is application. We need to find things that are useful for patients, that patients can use to improve their lives. We need to find them as fast as possible and as safely as possible. And all of this stuff comes with the caveat. I mean, the technology is great, but it's not the be-all, end-all. What's really important is that the research community works together synergistically to find things that work for patients. So, now to the science. So, I don't know how many of you remember this from biology, but the kind of dogma of molecular biology is that you have substances called DNA, and that makes RNA, and that makes proteins. Now, DNA refers to your genes, and the study of DNA is genetics, and you heard a lot about that from Sharon. I want to focus today on RNA, which is where a lot of my research has been focused upon. So, within the genetic code are genes, and these genes can make RNA, which is ultimately then made into protein. Protein is kind of the building block of life. And if you think about it, your DNA is your molecular fingerprint. So the DNA in your hair cells is the same as in your blood, is the same as in your kidneys. And it's nice to look at DNA and look for differences among people from DNA, but it doesn't give you enough information. There's another level to it. So why is your hair cell different than your kidney cell, different than the cell in your blood? And one of the reasons is because those cells use the DNA differently. So they access the DNA differently and they do different things with it, and they express different genes. So when we start studying the level of RNA and even protein, we're studying what the cells are doing. And that gives us really powerful new information to what's going on inside of the cells. And so the strategy with gene expression profiling is kind of what I alluded to earlier. You take 27,000 genes and you look within condition A and condition B, and you see what the differences are, and then you put those pieces together into a meaningful story. So let me give you an example. So we conducted a study called the NARES Study, the Nasal RNA Epithelial Sampling Study. And if I learned anything from Peter Merkel, it's that you need a good acronym before you can do anything. And we spend a lot of time actually coming up with acronyms. So if you have GPA, you'll notice that you more than likely have involvement in your nose. And it's one of the first places where you can get disease. And because of that, we were wondering, well, wouldn't it make sense that maybe the disease starts in the nose, or there's something with the nose that's a portal of entry or a window into the disease? And so what we did is we took this brush, it's a cytochrome brush, and you can just swab somebody's nose. I know I've swabbed a few of your noses out there. And within the bristles of the brush, it collects cells. And those are our cells in which we're going to look at RNA, and we're going to look at genes. And so we took a group of patients with GPA. We also did this in eGPA in Church-Strauss. And we looked at comparators. And then we could try to figure out what's going on with all of these 25,000 plus genes. And you get these really pretty nice graphs out of this. This is called a heat map. And I'm going to teach you how to read a heat map in 30 seconds or less. So what you're seeing here is each row, let me see if the pointer works. Yeah. So each column is an individual patient. Each row is an individual gene out of those 27,000 genes we talked about. And this purple bar, this group, is the GPA patients. The yellow bar is the comparator group. And what you do when you look at a heat map, if you start seeing differences in genes, you'll start seeing blocks of colors like you see here. And so what we're seeing is if their genes are red, that means they're turned on, they're switched on in GPA. And if they're blue, it means they're turned off. And so from these 1,600 genes that we identified that were, out of 27,000, we identified 1,600 that were different. And then we can start looking through those genes and say, how are they related? What are they telling us? What are we learning that's new about the disease? And I'm not going to get too far into the results of this study, but just to say that we discovered networks of genes that were related to inflammation within the nose. And we're using these to help us understand the biology of what's going on. Previously, when we look at your nose with GPA, we're limited by what we can see. Now we can start to see inside of cells. And we, I think, discovered a couple of genes that I think are promising candidates that may have a role in the cause of this disease. And what you do with this kind of research is you generate ideas from it. Now we have a list of genes that we can focus in on, and we can do the next wave of experiments to test these. Now we can get more specific and go back to the old hypothesis model. And that's how these models kind of interact with one another. Additionally, it's really important when you participate in clinical trials, you may not be aware, but we try to get the most that we can out of every clinical trial we do. So we collect samples within these clinical trials that we use for biomarker discovery purposes, and I'll give you an example of that. So within the RAVE trial, this is the trial that looked at rituximab versus cyclophosphamide in GPA and MPA. We took blood samples at various time points during that trial, and now we have patients that have been well characterized throughout a trial, so we have really great clinical data, and we can link that to the blood sample data that we have. And we took samples from before patients were treated. We took samples from month 6 when most of these patients were in remission of disease, and we took samples from month 18 when it was the last point of follow-up for many of the patients. And then we can start to ask these questions, and these are the exciting questions. Is there a group of genes that predicts who's going to respond to treatment? So we can look in the blood before you get the treatment and then compare that to how you do through the trial. Is there a group of genes that predicts who will have disease relapse? So we can look at the genes when you're in remission and then compare it to the end of the trial and see those who relapsed versus those who didn't. What are the differences in the genes? And it's way, way too early to get into this, but I think the answer to both of these questions is yes. Another approach that we take within RAVE is looking at proteins. So I remember I said DNA makes RNA makes proteins. So proteins are great to look at, but they're challenging to study. And Paul Monik led a study where this is kind of a hybrid, but it's more of a hypothesis-based study where he picked out 28 genes that he thought, or 28 proteins that he thought were important, based on what the medical literature told us and based upon our knowledge of the disease. And he was able to look within the RAVE data and see if these proteins were able to distinguish patients with active vasculitis versus healthy controls, and patients during the active phase of disease versus during remission. And he was able to find that a lot of these proteins did distinguish between these two, and many of them did it quite well. And what these constitute are potentially new markers of disease activity, new ways to measure if the disease is active or not. And for us, it's a celebration because it may mean that we were able to get beyond the CRP and the SED rate. Another example of how to do this, and I'll do this quickly, is in the RAVE trial, we just took your blood, but there are other groups that are developing technologies to actually take the different cells within the blood. And remember, there are a lot of different cells within your blood, and to sort them. And Dr. Monik mentioned to you about T cells and B cells and neutrophils and eosinophils. Well, wouldn't it be nice if we captured each of those in purified populations and looked and saw what was happening inside those cells at the gene expression level? That's what this group did with, and what they found was within a cell called a CD8 T cell, it predicted what was going to happen to patients with ANCA vasculitis, with GPA or MPA. And so they found a set of genes, which is called a gene signature, that determined whether or not you would stay in remission or whether you'd have a lot of relapses. And what you can then start to do with that stuff is the application part. And so there's another omics for you to have in your mind, which is pharmacogenomics. So if you have a set of genes and you can measure it in patients, patient A has those genes turned on, patient B does not, and then we can start to tailor therapies. Well, maybe patient A needs therapy X and patient B needs therapy Y. And this is what's often referred to as personalized medicine or precision medicine. This has long been the promise, but I really think that we're on the cusp of starting to actually put these gene signatures into clinical trials and testing to see if this stuff works. And I've mentioned a lot about GPA and MPA, but this goes well beyond those ANCA diseases. And for all of you out there with other types of vasculitis, the message is that this technique can be applied to your disease. So we can sort blood cells and we can profile blood cells, we can profile tissue cells in any number of diseases and learn novel things about those diseases. What is the limiting step to this is patient participation, which is your job, is cost, which is who knows whose job that is. These cost about $400 per patient to do a gene expression profiling experiment. And then expertise, which is our job. We need to keep training young investigators that are interested in this kind of research. And it's demanding research. It's challenging statistically to accomplish this research. But I think that there is a wave of young people that are really interested in this. This is a list of all the VCRC biomarker studies that are going on or have been completed. I'll just put this up to show you that we're looking at markers in the blood in GPA and MPA. We're looking at eosinophil-related stuff in eGPA. We're looking at studies in giant cell arteritis and Takayasu's arteritis. And we're within the biomarker approach, we're looking for genetic stuff as well, which Sharon touched on, so I'll just leave it at that. And it doesn't have to be stuff you measure in the blood. It can be anything you can measure. So another possibility is PET scan. So within large vessel vasculitis, you can measure inflammation on a scan in the blood vessel. And that's another possibility as a biomarker. And then the one that really excites me is the microbiome. So you have more cells in your body that are non-human. They're bacteria, they're virus. And before, it was challenging to kind of measure these. But now we can use these same high throughput technologies. So we could take a swab from your nose or some of your, swab on your skin and profile thousands and thousands of different bacteria and see if they're there. And what we could potentially learn is, are there bacteria or viruses that are causing these diseases? And even cooler than that, are they interacting with our immune system in ways that is influencing how the disease progresses? I think this kind of research is coming. It's coming in other fields, and it's starting to happen in vasculitis. So the last thing I'll say is I greatly benefited from training within the Vasculitis Clinical Research Consortium. And really one of the missions of the VCRC is to train young investigators. And one of the responsibilities of those young investigators is to go out to new places and do new things and reach new people. So I trained at Boston University under Peter Merkel and then Paul Monack. And I just left and I went to the National Institutes of Health as of a couple months ago. And what I'd like to do there is start a clinical and translational research program in vasculitis. We have the capabilities and enormous resources to do things like what I'm talking to you about, to sort cells, to do gene expression profiling, to do novel imaging studies and PET scans, and the wherewithal to do them within collaboration with the VCRC. And so I encourage all of you, as this program gets going over the next few months, to think about coming and joining and participating. One of the nice things about the program is you don't have to necessarily be close to a VCRC expertise center. We could perhaps pull in patients from different areas of the country. And it really follows in a tradition that I'm quite proud of being part of. So the original life-saving medications for GPA, the use of cyclophosphamide, was first tested in the 1970s by Anthony Fauci. And then two people that are here today spent a lot of their career doing really great stuff at the NIH before going to Cleveland Clinic, and that's Gary Hoffman and Carol Langford. So I'm really, really excited to have the opportunity to kind of walk in their footsteps and to start doing vasculitis research again at the NIH. There hasn't been anyone there focusing on vasculitis in the last decade or so, so it's nice to hopefully try to bring that back. And with that, I'll acknowledge all the people that have contributed to this work. I would like to acknowledge the American College of Rheumatology. They gave me a physician scientist development grant, which is what was used to do the NARES study, and the Immune Tolerance Network, which funded a lot of the work related to the RAVE trial and the gene expression studies that we're doing within the RAVE trial. So I hope that you hear the message. If RNA and DNA and proteins washed over you, I hope what you really heard is the excitement of what we're doing and the fact that we need everybody to participate and to give time and effort because I think that we're right on the edge of seeing really great benefits from these kinds of approaches. So thanks.